What's going on, GeneGuard family? Welcome to another episode of the Biotech Startup Series. Today, I'm speaking with Jack O'Meara, the co-founder and CEO of Okra Bio, a genomics medicine company. Okra Bio's mission is to cure non-alcoholic fatty liver disease with the help of small interfering RNAs. So, stay tuned and learn more about the drug development pipeline and how they might get their drugs into clinical trials without testing on animals. Well, hi, Jack, and thanks for joining my show. It's great to have you here.
Thank you. Very glad to be here.
So, Jack, let's start with your background and a little bit about yourself and how you got to founding Okra Bio.
Yeah.
Just a story a little bit.
Cool. Yeah. I'm happy to share. So my background is originally biomedical engineering. I was a tissue engineering person working on tendon scaffolds. Tendon was my kind of tissue of interest. But one of the things I was frustrated with while I was doing my kind of early academic work was that a lot of the science we were developing didn't have a lot of clinical application. The synthetic graphs we were building, clinicians didn't really like. So I was quite frustrated that a whole lab and a whole tens to hundreds of people were working on a technology that was unlikely to ever be clinically useful. So it kind of got me asking the question, what's going on here? So I then retrained and went to the US and did a grad school there, focused on how to bring new technologies to market with a particular focus on healthcare technologies. So spent a few years then after that really working in that early stage healthcare space, working with companies who are trying to bring new products to market and trying to get a better understanding of how the healthcare ecosystem works and all the very strange complexities of the US healthcare ecosystem and reimbursement and IP and all the kind of logistics that goes into it. So I really loved the kind of period between new innovation to actual clinical use and patient benefit, and I was kind of determined to figure out that whole spectrum of work. So that's what I spent a few years doing. Ultimately ended up at a gene therapy startup called Avexis, which brought a new AAV gene therapy to the market for patients, for children who had a rare neuromusculoskeletal disease called SMA, and that was a hugely inspiring project to be a part of, because just the clinical impact was unbelievable.
It was major.
Major. So.
Yeah, I've heard of that. Heard of that, and it's pretty amazing. Yeah, it's a single dose, right?
It's a single dose. It's a single dose. So one dose shot, and it's we obviously don't have long-term data on it yet, but it seems to be almost curative for patients who receive it. So we were actually just got approved in the UK by the NHS today. So but even though I know, kind of crazy, because I worked on it a couple of years ago, but I remember just being super inspired because as we were doing some of the trials, we would be putting together packages to send out to Japan or wherever we were sending product to, but ultimately every package you sent is saving a child's life. So it's just one of these kind of surreal kind of things to be a part of, and that really got me inspired and excited about advanced therapeutics and the power of a lot of these new technologies to really impact clinical disease and improve patient lives. So I came back to the UK. I'm originally from Ireland. I'm actually calling you from Ireland at the moment. It's a lovely day, which is unusual for this part of the world. But I wanted to get into the early stage healthcare space, which is kind of my love. And I met this guy called Quinn, who unfortunately couldn't join today, but is just a fascinating and incredible scientist with a deep background in liver biology. So he started off life. I'll give his background maybe in brief because he's not here. He started off life as a doctor working the wards, but then retrained really in kind of computational biology and systems genomics, and really wanted to understand the kind of elements of biology that we could try and treat. So he then went on to lead Oxford University Single Cell Genomics Consortium, really building a career in that field, and then ultimately Novo Nordisk's NASH liver program. He was head of genomics there, helping to work on this big problem that's facing the liver space, which is NASH and NAFLD and too much fat in the liver and all the cardiometabolic downstream effects of that. So when I met him, he was actually just wrapping up his role there. I was interested in pulling together a similar type of technological approach, but applying it to this problem of transplant, where a lot of donor livers now, people don't realize this, but they have too much fat on them, so they can't actually be used. So over the next few years, it's been really spiraling out of control because of an increased prevalence of fatty liver disease in the general population. So, and particularly older people. So more and more donor livers that come into centers have this problem with too much fat on them. So anyway, go ahead.
And is this, I just meant to ask you, is this like, I read that it's like, 25% of the adult population has this? Is this like across age groups, or is this like even higher in older?
It's definitely an age. It's definitely an age-associated disease, and that it gets increasingly worse the older you get. And as people get older, our metabolism slows down. We get kind of love handles that we just can't get rid of anymore. Well, the same thing essentially happens to your liver, where fat builds up in and around the liver cells, and it starts to cause all the problems with metabolism. And the liver is really your kind of brain or your metabolic engine. It helps you break down foods, but that starts to dysregulate, and ultimately that it will have caused the liver to fail. NASH is now the main reason for a liver transplant, but more broadly, it has a large effect on your cardiometabolic profile, and you're more high risk of a heart attack, and a lot of downstream effects. So maybe just the last thing I'll say, Quinn kind of gave me this pitch, told me all about the liver, taught me all about the biology. I was enamored with the idea of solving this problem in transplant. But it was quite funny how we first met, because I just left the US. I'd gone actually to spend some time working in Tanzania, where I've got a friend over there who does development work, and he had actually just left his job at Novo to go set up a treehouse in Costa Rica. So we were calling each other, and we were just sharing stories of like the wildlife we'd come across in these different funny parts of the world, and hadn't actually talked about our kind of shared love of advanced therapeutics until we met in London a couple of weeks later. But just quite funny. I think Quinn and I have a very philosophical alignment beyond our kind of complementary interests in healthcare technology.
Yeah, that's great. I think it's one of the most important things when being a co-founder.
I couldn't agree more.
That type of alignment is absolutely necessary. Okay, so you mentioned therapeutics for the liver, or fatty liver specifically. Now, can you tell me a little bit more about how you're approaching this? I mean, what's your research process?
Yeah.
How do you go about that? How do you do this target discovery? And first of all, what's the biology behind this therapeutics that you're trying to develop?
Absolutely, lots of great questions in there. I guess maybe just start off with the last one.
Sorry.
It's good.
Machine.
Keep them coming. I like the fast pace, but the two areas of biology we're particularly interested in are metabolism, so how the liver metabolizes and break down fats, etc., and also metabolic stress. So how, as the liver begins to build up with stress, how it can resist the urges to fail and the urges to call in cell types that cause apoptosis, which is essentially the failure in the beginning of kind of cirrhosis or earlier stage fibrosis. So anyway, we're particularly interested in those two areas of biology, and they typically play out earlier in the disease process of NASH. But the initial indication we're working on is this unusual one of all these donor livers that have a similar problem in that they have too much fat on them. It's primarily earlier stage disease, so before F1, which is the terms used to describe stages of disease in NASH. But they can't be used because they have a very bad response once they go into a patient post-transplant. They're very prone to having an ischemic response, which essentially it is this second bucket of biology, in that they have a lot of metabolic stress and they ultimately fail and have the really high rates of what's called early allograft dysfunction, or it basically means after the transplant they fail for patients. So we're developing a therapy that will be used to dose these livers ex vivo, so outside the body. Actually, we put them on a machine to keep them alive outside the body, and we then administer this therapy.
That's pretty amazing. I mean, you got to talk about that a little bit.
Okay, sorry.
Once you finish your idea.
I'll just get out. Maybe I'll get out the kind of piece about our broader strategy, and then I'll come back to the machine. Because the machine is pretty cool. I got to say a bit more about that. But so they're dosed ex vivo, and then the therapeutic that we use is a GalNAc siRNA, which is a gene silencing therapy that plays out for many months, so up to 6 months. So then the liver goes back into the patient, and the therapy we administer helps to basically increase its resilience to metabolic stress, so it doesn't have this ischemic response and fail, but also increase the metabolism. So over the course of the treatment, over the course of the 6 months that this therapeutic compound is active, it's breaking down the fat and helping to improve its actual fatty liver disease profile. So maybe the last thing I'll say before I go back to the machine, because I definitely want to get back to the machine, is that we see this as a novel way to rethink NASH, which is a very big challenge and a big problem that a lot of pharmaceuticals are working on, because NASH is very difficult to design drugs for a few reasons, and to maybe oversimplify a bit, but there's no good preclinical models. So all the mice models, there's tons of different mice models, none of them are very representative, none of them predict how a drug is going to perform in the clinic very well. And then secondly, when you go to the clinic, it's very difficult to design a clinical trial because patients don't actually know they have NASH until they very late stage NASH.
Right. Because it doesn't hurt.
It doesn't hurt. You don't have any
symptoms.
In their
you feel day-to-day activities.
But then all of a sudden you get very severe symptoms, and that's because the disease has actually progressed over the course of 10, 15, maybe 20 years. You've been continuing to build up this fat, and it's caused all these problems. And then at that point, it's very hard to treat because it's so late stage and so far gone. So we think focusing on this problem in transplant, where we're studying earlier stages of the disease, because they're not cirrhotic livers, they're earlier stages, we can better understand the biology, better intervene, and prove that our therapies in this transplant context can actually have broader applicability to patients who may be suffering from NASH and NAFLD. Anyway, long-winded answer to your question, but maybe to kind of circle back and kind of try and tie it together with the machine and our drug development process. So I'll give you a little bit of background on the process, how we think about developing these GalNAc siRNAs, which is a kind of technical term for basically a gene silencing therapy, which has the ability to cure or positively affect the disease. So our drug development pipeline broadly matches to a traditional pharmaceutical pipeline, in the sense that we have a target discovery group, we have a target validation and lead optimization group, and then we have a kind of a preclinical work. The difference, we have a few different differences. One is a machine, and I'll get to that on the kind of the latter stages, but from a target discovery perspective, we are heavily genomics focused. So we use a lot of the most advanced genomic technologies to really tease apart disease, particularly chronic and complicated diseases like fatty liver that plays out over multiple years. So one of our first big target discovery programs was a partnership with Oxford University, where we took 1,000 liver samples. These are tissue samples of every liver that was transplanted in the UK in 2017 and 2018. So these livers are walking around in people out there, and we took these samples. We did a very advanced genomic analysis in them, where we imaged them multiple times at multiple resolutions, and we applied an imaging AI technology to basically standardize what's called the histopathology, or basically standardize what the disease state was that these patients had. We also used imaging AI to try and annotate different sections of disease within the same tissue. That might sound a little bit complicated, and maybe to make it simpler, basically we use this technology to identify regions within a tissue that's healthy, and then compare that to regions that have slight, what's called hepatocellular ballooning, but basically what it means is early stage disease, and then compare that to inflamed and later stages of disease within the same tissue, because this disease is very heterogeneous, meaning there's a right next to different sections right next to each other have quite different states of the disease. Exactly. So then we image them, and we used imaging AI to annotate different stages of the disease, and then we took cells using a kind of a special technology to take out cells of those particular regions, and then we sequence them. So what that allows us to do is to ask the question, what's going wrong at a genetic level at the earliest stages of the disease that's leading to these changes, that's getting more inflamed, and then ultimately it's getting necrotic or actually quite disease? And what are the genetic signatures?
Just a question regarding the
sequencing part. Are you also doing DNA or just
RNA? So are you interested in the transcriptome?
So we're primarily interested in the transcriptome and how the transcriptome changes at different disease stages, but we also do some genetic work just to get a baseline. But we did have a lot of difficulty with these samples because they were quite tough samples to get good DNA work done, but we are interested in kind of understanding the genetic baseline as well, so we can better compare our results, but primarily RNA.
Okay.
So once we've completed this large study, we did that same type of analysis that I just described on one tissue section across 1,000 patients, and then matched that to 150 clinical variables. So how those patients' BMI was, how those patients performed in transplant, how their bloods looked over the time period before and after the transplant itself. So really building this robust and rich data set to better understand disease progression. And that's a key feature of this disease, because it plays out for so many years. It's what is happening at that earlier stages that's leading to later stages of the disease. And people, different patients have different responses. So if you can kind of better understand that question, you can better design therapies that will be more effective at intervening and preventing and protecting patients against very late stage, what ultimately transpires in patients needing liver transplants and all those awful challenging conditions.
Got it.
So long-winded first introduction, but that's our target discovery work. And when we actually just finished that large study, and now have a list of about 250 very interesting targets that we're progressing into our next stage of development, which is our target validation work. And at this stage, we've got a team out of Oxford, out of the bioescalator there, who are doing amazing work. And we've got a really wonderful group of people coming together where they will work on testing out siRNAs. I've been using that term a little bit. It basically means a gene silencing therapy. So once we know the genetic signal, or the transcriptomic signal, I should say, so we know the RNA we want to attack or knock down for the benefit of the patients, we then test out siRNAs, which are basically they're basically silencing RNAs. That's not the actual term, but they go and knock down the RNA of interest, and then we monitor if we knock down this gene, how does that affect the physiology of the cells, of the liver cells? And we do these.
And are you doing this transcript by transcript, or I'm guessing that's your approach,
right?
Yeah. We
so take just one transcript by transcript.
Exactly.
See what happens.
Exactly. We've identified about 250 genes we think are really interesting and really important to this disease. So we'll go through screening those using a basic 2D tissue culture model. It's a 2-week model, and it recapitulates some of the transplant stuff. And then we move it into more advanced co-culture, so multiple cell types in the same model, to test out if our kind of initial theories holds true. And then ultimately we go through and test it out in precision slices of liver. So these are little liver slices that we keep alive after they've been taken out of the liver, and then test out the therapies in those as well. So we're really trying to get coherence and prove that the insights we've gotten from our genomics-based work hold true when we test out siRNAs in multiple models. And then the last step of our development is, at this point now, we've got pretty high conviction over a number of genes or what I call siRNA leads that we think are really, really impactful and could be potentially very impactful for patients. And then we go and we optimize a GalNAc siRNA. So this is basically a little sugar we stick onto the siRNA, which is the medicine, which is the kind of small interfering RNA, and that sugar makes it very liver specific. So it means the hepatocytes will just soak it up and really, really absorb it quickly, and it means that the actual siRNA will be as effective as possible for the patient. So we optimize our own GalNAc siRNA, and we perfuse that in a whole human organ kept alive outside the body. So let me explain in a little bit more detail, we work with transplant centers both in the UK and internationally, and now we're setting up a site in Canada, and we'll have a few in the US, to work on these discarded donor organs. So these organs that have been rejected for transplant because they have too much fat on them, and surgeons don't feel comfortable using them. So we work on them to test out the therapies that we're developing to solve the same problem of fatty donor livers. So we hook these livers up to the machine. That's where I get to go talk about the cool machine we work on, and we test out the actual therapies we developed, and keep the liver alive for multiple days, so that we can see if the therapies we're developing are actually improving the function of the liver, and therefore will improve the function of the liver once it goes into a patient. Now, we haven't done anything like that yet, but we're doing all of the preclinical work on these discarded donor livers that don't go into patients to get enough confidence and proof that what we're developing will actually meaningfully impact patients when it does.
Got it. Now,
just getting back a little bit to the siRNA, how are you looking to find one target? So is this going to be like a one target? Do you think this is a monogenic type of disease, or is there a complex of genes that are have this complex interaction that you're going to have to knock down or reduce in some measure?
Yeah, that's it.
I think this is one of the, as far as I can tell, is this one of your challenges?
Can you
absolutely.
Talk about that
happy to answer
and
that.
how you're trying to figure this out and overcome this obstacle?
Absolutely. Great. That is a great question, and it's super pertinent to the field right now, because for chronic complex diseases like NAFLD, like NASH, and that's the clinical term for fatty liver, that it's highly unlikely that you're going to find a silver bullet where it's a one gene therapy.
Right.
And we philosophically from the beginning have been very focused on figuring out combinations of targets that we think would really affect the physiology. And there's multiple things going wrong in NASH, because it's been building over a long time. So there's multiple different pathways that one needs to think about. And that's why we're so excited about our modality, GalNAc siRNA, because you can attach multiple siRNA, so multiple gene targets to the one GalNAc, and it becomes a combination therapy, but in a single molecule. So we are
molecule. That's cool.
Yeah. And the other thing, because we've been heavily genomics led, so I talked a little bit about our target discovery and all the genomics work that went into that, but equally at every step of our pipeline, so all of the 2D models, all the 3D models, all the liver work we do, we sequence all the time. So we sequence all of that data to really understand when we're hitting this gene, how's that affecting the rest of the transcript, and what will be interesting combinations, and how would those combinations affect the rest of the transcript, ultimately to get to that healthy transcript that so many patients have. So we are very focused on using a lot of these new computational technologies to understand and tease apart the biology using genomic data to really figure out what is the right, but the most powerful monotherapy, but also the more powerful combinations of siRNA.
Combination, yeah.
Okay, now once you get to the actual ex vivo livers that are kept alive, first of all, one question regarding that: how long are they viable? Is it a matter of months, years?
No, definitely not. So a liver in clinical practice can only be kept alive, can only be outside the body for a day before it gets transplanted. So this is because traditionally in clinical practice they were put on ice for a long time, or they were always put on ice. So they're very hesitant to use a liver that's been outside the body for more than a day. Now, along come these machines. So these perfusion devices are a very new technology in the transplant world. Well, I say that they're new and they're old, and that they had a run in the 90s, but they didn't work very well, so no one used them. And now they're re-emerging as a very novel way, because they're a lot more advanced technologies. But anyway, they are used to keep the liver alive. So basically, you kind of imagine a box where you put the liver in, and it pumps it with blood and oxygen and nutrients, and basically just reimagines what the liver would feel like if it was in a human body to keep it alive. It keeps it warm and all of those kind of key things for the liver. So experimentally for us, as we work on developing the best possible therapy for patients, we will keep the liver alive for up to 5 days. If we can go longer, we will. We're working with some groups who have done it for much longer, but our goal now experimentally is to consistently keep these livers alive for five-ish days at a time. Now, to date, we haven't done that, but that's where we're moving towards, and ultimately, hopefully, longer as the technology evolves. And as we test out our therapy, we'll be taking samples and sequencing and assessing how that liver is performing over the course of that time, and a better understanding how our therapies are ultimately affecting the physiology of the.
Okay, got it. So it's a pretty short time fuse.
Yeah.
So you have to be able to. So the effect needs to manifest within those 5 days for you to measure it and understand that you're on the right path, because you're not gonna. There's not going to be any visual changes.
No, we're not trying to massively defat the liver in a short period of time. Now we do take cells from the liver and culture them for weeks after the experiment. So we have up to, I think now we're looking at a very long couple of month time frame where we can keep cells alive from those experiments. So we can keep cells alive and assess how the physiology is changing in the cells. But really we're trying to understand PK/PD in the liver, how transcriptomically how the liver is changing over the course of the therapeutic administration, and then we'll keep cells for a life much longer.
Got it. And once you get there and you identify a few targets that seem to do the trick and seem to push the data in the right direction, what's next for you? Can you directly go into the clinic, or do you still have to go through some animal models, or how does this work from a regulatory point of view?
Yes, it's a very kind of timely question. We're just thinking about all of our all of the non-clinical work we need to go and do, and planning meetings with the agencies as we speak. So I don't have a great answer for you at this point. What I can say is
it's still uncharted there.
It's still uncharted
because you're testing on a human organ, but that's kept alive ex vivo. So it's better than a rat, for example. That's usually the model that's being used, but still it's not an organism. So it's tough to say whether it's safe to go into the clinic directly. So
exactly. It is a very new kind of space. The one thing to note is, because we're using these GalNAc siRNAs, is that they get soaked up into the hepatocytes very quickly. So, and then we monitor the livers thereafter to check if there's systemic effects. So if more GalNAc siRNA is coming out, and there's minimal systemic effect of our therapeutic dosing. So that creates more questions about well, if the therapy is in the liver and it never leaves the liver, what does that mean for when the liver goes into the patient? And you're kind of in the realm of cell therapy where you've got kind of an organ therapy. So the organ is the medicine for the patient who has all these ailments.
Right.
So it's a very interesting regulatory question, and one we are building our hypotheses internally for, and we'll also be working very closely with the regulators to make sure that we do everything to keep patient safety top of mind. So lots of work to do.
Mm-hmm.
Yeah, definitely. Okay, now getting back to the business part of things. So we understand the technology. It's super interesting. Now, how did you get to this point? Because you're pretty advanced in terms of your pipeline. You have a pretty much established pipeline. You have data. You're gathering data. How did you survive for so long? I mean, I think this is one of the biggest problems for early companies. They don't have funding. They have a dream, and that's pretty much it.
Yeah.
What are some of the lessons that you've learned across this period of time, and that you'd like to share with future founders?
Yeah, no, I the valley of death is the biggest challenge for a lot of early stage, particularly biotech companies, because it costs so much to get going, which isn't necessarily which isn't necessarily the same problem that tech has. So I think some of the things we really focused on to get started. Now, there's obviously routes like grant routes and all of that. That's very available, particularly in Europe, which is one of the good things about Europe. We actually were very focused on private funding because we really wanted to move as quickly as possible. But so I'll talk to you primarily about how we thought about building enough credibility to attract venture capital funding or even angel investment. I think that was the route that we decided to go down pretty intentionally early on. So as we thought about that, really, as you kind of alluded to, once it was me and Quinn, you're two folks and a dream and a written on the back of a napkin, kind of your plan for what you're going to do, and a lot of wind to back it up. But one of the things we really focused on was how do we make sure we have a very credible story that investors will take seriously. So some of the things you can do to bulk up your credibility is you can find fantastic advisors. So you can look up who in the world is the world expert in the area of biology that you are focused on, and we were very intentional about finding people who could give us that level of credibility that maybe we didn't have, at least initially in the area of biology we were focused on. Number two is you can attach yourself to really prestigious or well regarded accelerator programs. So we actually, I think highly unusually, did 3 accelerator programs simultaneously. Again, very, very, and I wouldn't do it again if I was maybe starting over, but it was through this, it was kind of with the idea being we were hell bent on getting attracting investor interest, and one of the best ways to do that, I think, is to have multiple brand name kind of accelerators on your back. So we started off doing a program in London called Entrepreneur First. That was actually where we initially got connected to, and that got us going. And then we were thinking more about where we started talking to European investors and found it's a little bit slower paced than we maybe had hoped, and it was a little bit more difficult to raise than we thought we were going to have. So we quickly then started to look over to the west coast of California, where money flows a lot more freely and you don't spend months on end talking to investors. You can get going quite quickly. So we looked up a Y Combinator, and we were lucky enough to get selected to participate in the Y Combinator program, which was a wonderful wonderful experience first off, but also was an amazing asset from a fundraising perspective, because now you've got a brand name you're associated to, but also you've got this amazing day, this demo day, where investors are all eyes on the companies, and you can kind of create this competitive tension between investors. And we did that quite well in a respect, in that we had all the European investors we've been speaking to, and now we have Californian investors, and we can kind of create a bit of competitive tension between the two, which tends to help the
That's pretty smart.
Yeah.
Obviously. Yeah.
And then
you want a bidding war.
I mean, it's the same in any negotiation. You want optionality. If you're relying on one person, their job is to make money for their fund. So they're obviously going to want to pay the lowest price that they have to pay to get the deal. So if you've got more people competing, it's always going to benefit the founder. And the last program, I mentioned that we did was a program out of Europe, which I actually found really helpful, called Gold Track. And what Gold Track is, it's a European initiative, but they've brought together some of the really premier life sciences investors and executives from around the world, but those maybe with a kind of an interest in Europe, and you get very hands-on kind of exposure to them, and that really helped us again because we got to build our advisory board. We added some the CSO from Novartis Canada to our advisory board. She was amazing and kind of helping us get started, and we also got one of our investors who was previously president of SR1 for a decade through that network. So again, it was trying to attach ourselves to people who had a lot of industry credibility and able to for investors to be able to talk to and say, yeah, those guys they're the real deal. They're not just a load of wind. They actually have a very credible plan. So those are some of the things that we thought about in terms of how we got going, and then just leveraging everyone and anyone that in the investor community. And I know that's kind of a difficult one because people aren't necessarily tied into it, but neither was I, quite honestly, until I kind of found my way into those networks through EF and Y Combinator and these type of programs. So I hope that's helpful for some folks in the audience.
Yeah, definitely. But at the point of fundraising,
yes,
did you have something?
Did you already have something?
Or it was just like a good pitch deck, like a good background, some experience in the field.
Yeah. So that's a question we get asked a lot. I think it's a really great one, because a lot of people will ultimately, when you're that early, no one has anything. I mean, you have some really powerful IP, but if you're a very early drug discovery company, it's unlikely that you'll have you'll have your final product, I guess, unless you actually have a IP around a particular compound. But for us, because we wanted to essentially set up a fully integrated biopharmaceutical company from target discovery to validation to preclinical, we wanted to set up a full stack pharmaceutical company. Whatever we had at that early stage of our development was going to be really minimal versus a year and a half later where we are now. So what we had was what we thought was sufficient evidence to go out and tell a story to the market that says, look what we've done with 150k. Imagine if you gave us X that, what we could go and do with that extra capital. So what we essentially done was we generated data on each piece of our pipeline. We generated some sequencing data on a number of livers. We generated some in vitro data to show that we could run in vitro experiments, and we generated some data on actual precision slices of liver. And basically what we said was, with in 3 months and 150k budget or 200k, whatever the budget was, we've been able to fully operationalize our whole pipeline. Now, none of the data that we generated from the target discovery there wasn't the full flow through that I've just described in our current pipeline, but it was enough for investors to get conviction that, okay, in a really short period of time, these guys have been able to achieve a lot. If I invest in these people who I believe are go-getters and are going to actually make shit happen, excuse my French, that they will be able to go and do that. And I think that's sometimes where I talk to founders and they're kind of bogged down. They're like, I just don't have enough data. No one's going to take me seriously, and I don't have enough data. And it's obviously very case specific, but I would urge you that it's not always about having the most data and having a really tight package that you can bring. It oftentimes is about what you've achieved in the period of time you've been working on it. And if you can demonstrate having just achieved a lot and having really proven out your thesis and can give conviction to someone who's betting primarily on you more than your IP or more than your technology at that early stage, that you, given the resources, you can go and really hit the gas pedal. I think that's often more powerful than having a super robust data package, would be my comment.
Okay, that makes perfect sense, and that leads me to my second question. In terms of investors, there are multiple types of investors, clearly, right? So you have like the Silicon Valley type of tech, trigger happy kind of investors that, they look at buzzwords such as crypto, AI, space, hard tech, whatever the buzzwords. And then you have like the white collar suit kind of investors that are, old school, but they have different views on how to approach this. So for a biotech company that's sort of like in between. So nowadays, it's not just lab work; it's clearly computational biology in a great measure. Anyway, what type of mix of investors do you recommend people go after?
That also is a great question, and to some extent company specific. But I can tell you a little bit about how we thought about it, because we definitely are kind of squarely in that intersection. We have a traditional wet lab, but ultimately, all of our genomic data is feeding into algorithms that we build ourselves and into data sets that we curate ourselves. So in that respect, because we recognized how much we sat in the middle, we did put emphasis on finding kind of essentially archetypes from each camp. So we identified a guy who I mentioned earlier from previously from SR1, now in a group called Apollo, who is a very traditional therapeutics investor, really sharp on the clinical development, the medical side, all of the work that's needed to bring a medicine to the clinic. And then equally, we landed on a Californian investor who we're really big fans of, who's very, very sharp. Oh, let me turn off my. Sorry now about my.
No problem.
WhatsApp. But anyway, who's very sharp on the genomic side, on kind of the computational biology side of the house, and we tried to marry that mix together, to because it's important. And also we have a tech, kind of a similarly comp biotech investor out of London who's closer to home and could help us kind of with the building the business piece of things as well. So we really try to straddle the two of those worlds, and equally we're a European company with a US top parent company, so we try to get US investor with European base as well. So those are some of the philosophies I think are important. And maybe some comments on that is, oftentimes when you're very early, you'll have more luck on the West Coast because they'll just move quicker, and there's kind of an old school mentality to a lot of the traditional biopharma investors, where they're kind of looking at you like, who are these guys? It's a bit more of a closed cottage industry as well. So what at least our thesis is, we do need, we're fully intentional about finding traditional therapeutics investors to kind of double down on us and help expand that, particularly as we now look towards the clinical development. But equally, if you can get started and go and generate the data you need to really excite those traditional folks and really get them going, then that's a good strategy. And that was actually one of the things some of the East Coast investors I was speaking with were like, we like it, we think it's innovative, but we think you're better off going getting money elsewhere and then come back to us and we'll pay the extra price at the A, because that's generally our sweet spot anyway. So it's a balance.
Right. They trade money for risk, basically.
Basically.
That's what they do.
Yeah.
Right. Okay, Jack. These were great insights. Thank you very much. I think we covered pretty much everything. So it was great having you. Thanks again.
Thank you. I'm glad. I hope so. And if anyone wants to reach out to me, you can get me on Twitter or otherwise you can find me somewhere.
Yeah, I'll post everything in the description, and maybe we do another episode in, I don't know, 6 months or a year, just to assess the progress, see where you guys stand at that point.
Love that idea.
Super interesting.
Love that idea. And I'll bring Quinn. I'll bring Quinn next time. So we'll have the
definitely. We can drill down into the AI, the hard biology.
Absolutely. That sounds great.
Okay. Cool. Thanks again, Jack.
No worries. Thank you for your time.
See ya. Bye-bye.
Bye-bye.
This concludes our episode. If you liked it, hit the like button, subscribe, and follow me on Clubhouse @RaduTanasa. If you need an invite for Clubhouse, just drop me a line in the comments and I'll send you one. See you soon.